So I'm going to continue with applications of random knotting to analysis of DNA molecules. But before that, I continue with the homework that Radmila assigned, which was looking for knots in Freiburg. We found this one that is not quite a knot, but it kind of it's an interesting knot. And then we found this knotoid next to the movie theaters. So it's up to you guys figure out whether this is actually knotted or not. It looks like. All right. So so as I said, I had originally proposed something a little bit different to talk about bacteriophage one day and trypanosomes the second day. But I thought that this is a new technique. It was introduced after the genome was, or the human genome was sequenced, and it makes heavy use of sequencing of the genome. And basically what it does is use, and I'll explain the details later, it uses the sequences of human genome to map regions in 3 dimensions and kind of provide reconstruction for genomes. And these techniques overall are called chromosome conformation capture data. And they come in different flavors. Some of them are called 3C, 4C, high C, 5C. So there are a number of them, and I'll be explaining some of those. All right. So just before we move forward, let me kind of review what we did last time. So basically, we have a biological problem that we are interested in analyzing. And what we do is we come up with a polymer model or a DNA model that we think is suitable for the questions we want to answer. And from that model, we do either simulations, and when possible, we try to develop analytical results. And that's, to some extent, straightforward. The hardest part is usually kind of coming back from the theory to the biology. So what I always recommend is that we have to be extremely cautious, correct? We can come up with results that look promising in biology or either in computer simulations or in analytical results, but then going from there back to the biology and claim that something is happening in biology and it's a general feature, that's a big step. And we usually said, well, our results suggest that this might be happening, right? But biology tends to be very complicated, and a lot more factors can be playing roles. And so we have to be careful. Of course, you always have the option to keep developing some more math, which is also very interesting. So usually you start with a biological problem, you come to a math problem, and this math problem kind of fits itself and comes up with new mathematical questions. I think the POTS model that was discussed yesterday, I think was a very good example, right? On how you can keep producing more math from an apparently applicable, or from a, in this case, physically motivated problem. All right, so just kind of continue our review. Remember that the first model we look at was a random knotting of DNA molecules in free solution. And what we said is that if you have a molecule that is long enough in solution, then it will form a closed molecule, provided that you have some sort of sticky ends or complementary ends. And we can estimate what's the probability of knot formation in those cases. And that was used in these 2 studies to estimate how thick the DNA is. So remember that the DNA is a negatively charged molecule, and because it has negative charges along the backbones, and depending on, or the volume of this cylinder, if you want, will depend on the ionic concentration in the media. So if we put more counter ions, more positive ions, then you can consider that this molecule is effectively thinner, and if you put less, then it'll be thicker. And whether how thin or how thick it is will determine the knotting probability of that molecule. So that was what was used in these particular studies. All right, so we also said that these in vitro results, so these DNA molecules becoming cyclic in solution, could be at least partly explained by those two theorems due to Janan Diao that tell you what's the probability that a curve makes a knot. And we said that this probability grows exponentially fast with the length of the polygon. And also we can ask what's the complexity of the knot measured by the average crossing number. And remember that the average crossing number is not quite telling you what type of knot it is, but it's telling you the average number of crossings that you will see when you take multiple projections. And it tells you that it grows quite slower than the knotting probability. So the chances that you have a knot are high, but the complexity of the knot will tend to be relatively simple. Now again, this is the math. If we want to go to the biology, we will have to make extra work. So this is giving us kind of a qualitative information of the process. But if we want to be really predictive about the process, then we have to do extra work. And I think that's still, to some extent, open question. So there has been work doing simulations. As I mentioned, the worm light chain. You can use the worm light chain to do some of these estimates. But can we analytically develop models that predict the observations in the experiments? That is still an open question. All right, so then we talk about this problem of DNA packing in bacteriophages. Remember that the bacteriophage is this virus that infects bacterium. It has this head called the capsid, and the DNA is packed inside the capsid at a very high pressure. What it makes this high pressure is three factors. One is that the diameter of this capsid is about the same length as the persistent length of the DNA molecule. The persistent length is the length of the DNA molecule under which DNA behaves like a rigid rod. So you are trying to really flex this molecule inside a small volume. Then of course you have DNA repulsion due to the negative charges that I mentioned earlier. And then you have again loss of entropy when you pack your DNA in such a small volume. And then we say that for a particular type of viruses, you can extract the knots, and then you can send them in a gel, and then you have this nice knot distribution. And as I mentioned, these knots are just the first. This is less than 10% of the knots that we see. It's probably like 5 to 8%. Most of the knots are a lot more complicated, and we don't have a way to know which knots they are. We don't have a way to understand if they have any information. So all those, again, are open questions. We don't even know whether this is what type of knot that one is. So if you see here, this is the knot. This is a trefoil, the 4-crossing knot, the 5, and we see that the 5 is a toroidal, because if it was a twist, it will migrate a little bit farther. Then we have 6s, and we believe, so this could be the prime. This might be the connected sum of two trefoils, but we don't know yet. All right, and presumably the distribution of these knots are going to tell us, or going to give us information about how DNA packs inside these viruses. We also talk about this organism, the T. brucei, which is responsible. T. brucei is responsible for sleeping sickness disease in Africa, and T. cruzi is responsible for Chagas disease in the Americas. And basically, this is the mitochondrium. When you extract the DNA from the mitochondrium, it's made of a large network. Remember that these are topological networks, meaning you have subunits and they are topologically linked rather than chemically linked. And the estimates back from the 95, this is from our review, but the estimates tell you that every DNA molecule is going to be linked to 3 other molecules on average, and the linkage is going to be just a Hopf link. It's going to be a trivial link. The way this structure was uncovered was first kind of looking at this pairs of molecules by cryo-EM, and then by kind of breaking this structure into pieces, and then as is shown in here, and then when you break this structure, you look at the frequency and the type of links that you get here, and based on this frequency of links, then you try to infer what is the general structure of the network. And again, this was work done in the 90s, mid-90s, and that's something we are trying to reproduce at the moment. All right, so we introduced this this algorithm to sample DNA molecules, or in general polymer molecules, in confined volumes. We said that this is the freely joined chain. It's a model that is fairly good for modeling DNA and other type of polymers, and there are algorithms that tell you how to sample the space of conformations of these polygons. Of course, the problem comes when you try to fit these molecules tend to be fairly expanded. So when you try to fit these molecules into a confined volume, then you have to come up with tricks. And the tricks that people use could be either Metropolis Monte Carlo or multiple Markov chains and so on. But again, this is still a very interesting problem. And I said that there is no theorem for, well, we can do simulations. And I said that this was still an open problem, and there are a couple of issues here on why this is a problem, correct? One is because, if you remember, we said that if we have a random walk in space without confinement, we know what the distribution of vertices is going to be. So for a given distance, or if you know the vertex k, I know in probability where that vertex k is going to be located with respect to the origin. That simple question is unknown for something that is confined. And furthermore, one of the problems, I don't know if this can be seen well, but if you have this is part of the proof of showing that the knotting probability of a polygon in free solution is going to be very large, grows exponentially fast. And the idea is that if you have a polygon in free solution, you start, this is the origin, first vertex, second vertex can be kind of anywhere in this sphere, and then you can make this structure here that is going to make the knot. Now, what's the trick about this problem? So you have to, so you see here, this is a polygonal change, an equilateral random change. The problem here, and the reason why you have to take care of all these details, is because you can always have the chance of having your knot coming back and undoing anything you did before. All right, so you tie a knot, then there is certain probability that your random walk will come through again and will undo all the work that you did. Now that can be estimated as kind of shown in here for polygons in free space, but we don't know how to do for something that is in confined volume. The way you can think about it is just take a sphere and then take segments in a sphere connected with each other. How do you know that once you've made a knot, you won't have another segment undoing that? Again, that's an open question. So this naturally, I mean, these are basic problems, correct? Just a polygon inside a sphere. How do we estimate the knotting probability? So all those, as you see, can kind of pose very interesting mathematical problems. All right, so the conclusions from the first talk were that the topological complexity increases rapidly with confining volumes and also with the length of the DNA molecule. In general, you can substitute DNA by any polymer, linear polymer. And also the topological structures are going to be complex, just being driven by confinement. So biological systems tend to have solutions to decrease this, or well, I would say more than 10, they have to have solutions to decrease this complexity. Otherwise, things wouldn't work. There has been experimental studies that show that if you have a knot in a DNA molecule, then you won't be able to transcribe and you won't be able to do basic biological processes. So knots are things that the cell wants to avoid. And as Mariel was explaining, after DNA replication, when the cell is going to divide, you are going to have links, and the number of crossings of these links is going to be determined by the double helix. So at the time of division, you are going to have all these links that again need to be resolved. If those links are not resolved, by topoisomeresis, when the cell is trying to divide, eventually will break one of the DNA chains, and those breakage will induce cell death. And as Mariel explained, that's one of the methods how we treat certain diseases, all right, including bacterial infections or cancer. It's just because a pooling of these DNA molecules will introduce double strand breaks in the cell, in the DNA, and the cell can not repair that and dies. All right, so then the question is, to what extent does this apply to higher organisms? And this is where this chromosome conformation capture comes into place. So for many years, we knew kind of the two extremes or the two ends of chromosome organization. On one hand, we know how DNA, what I've been explaining is how DNA, naked DNA, falls in solution. And we know that for higher organisms, DNA, and even for bacteria, there are certain proteins called either histone or histone-like proteins in bacteria that serve to or help DNA to fall in 3 dimensions. So you want to condense the DNA molecule. Basically, because you have, Mariel explained this, you have these large segments of DNA that have to be compressed in a very tight volume. So one level of condensing this is through this histone or nucleosome formation. Then this forms into the 10 nanometers fiber, which is pretty well accepted, and then the 30 nanometers fiber, but these may depend on cell conditions and other biological factors. Now, we don't know what is beyond this point. All this is an open question. And then we know that at the end, the cell is organized into chromosomes, what are called chromosome territories. And the idea is that every chromosome occupies its own area or its own volume, as is shown in here, all right? So every color represents a different chromosomes, and because we have 2 copies of each chromosome, you should expect to see 2 spots of the same color in here in this fibroblast. This is a human cell. All right, so this is a technique that came up recently, called chromosome conformation capture. I would say that it started in the late 90s, and it really has improved or has really expanded in the last maybe 10 years. And the idea is the following. This is the technique called for high C, developed by Eres Lieberman and others at MIT. And the idea is the following. Imagine this is the genome of any organism, and you can cross-link these fibers. So basically, you can grab them together. Then what you can do is you add this enzyme called HindD3 that what it does is cut the genome at the specific sequences. So now you go from here to this fragment. So now you have all the genome fragmented into pieces with these kind of sticky ends at the end. Then what you can do is fill these ends with a specific marker called biotin, and you ligate them. So you basically seal them back together, as is shown in here. And these fragments will have this marker here, this biotin. All right, so once you close this, you can remove your cross-linker, whatever chemical you put to bring these 2 DNA molecules together. You can break it into pieces, and then you can recover the sections that have the marker. All right, those sections that have the biotin marker, you can recover them back. And now you know, by this process, you know that, or you have a sample of which chromosome was near to which chromosome, or which fiber was near to which fiber. And from here, because we know the sequence of the human genome, then we can say, well, this part, we can sequence these sections, and then we know that this part corresponds to 1 chromosome, and this part corresponds to another chromosome. Therefore, if I see this frequency or this type of readout very often, I would say that those sequences were were in close proximity for most cells, right? Of course, it's going to be an average over many cells. So, but what is a key aspect here is the idea of chromosome proximity. Things that are close in space, or chromosomes that are close in space, will have more DNA readouts than things that are far apart. All right, does that make sense? Yes? Yes or no? Yes, yes. All right, so the idea then is to look at these frequencies and try to make maps of the human genome, or any genome for that matter. All right, so there is a couple of measures that we can take out of these studies. One is what is called the contact probability. And basically, this is what I just explained, where you have these partners and regions that are near each other. They will interact more often than those that are far apart. So here in the x-axis, you can put the distance. Let's say if you are along the same chromosome, you can put the distance in base pairs. Here you can put how often they interact using this measure. And you can come up with a fit and a specific line here. And we're going to get back to this. This is one information. The other information is this one. You can produce these heat maps that tell you how often each region in the genome will produce or will be linked or will be near another region. So this is what you would observe in the experiment. Then you can say, well, this is what I observed, but what I observe might be biased by the experiment. So what I can do is have an estimate on what I should expect, and then divide observed values over expected values. And then you can start recovering a little bit more information, as is shown in here. And then you can do a little bit of normalization to clean this data, and this is kind of the type of data that you have. Now, you're mathematicians, and now you can start interpreting this data. So this is chromosome 14. This is the Q arm of chromosome 14, the long arm of this chromosome. And now you look at this. Red means that those two regions interact a lot. Blue means that those regions interact too little. The question is, can you fit a curve that corresponds to this pattern? So that's an open question, and people have tried different methods. It's, of course, the shorter the genome, the easier it is to solve, but it's still, again, a difficult problem. You can think like here, for instance, if you say, well, how does this little piece of the genome interact with the rest of the chromosome? Well, first, of course, regions that are nearby, excuse me, regions that are nearby will interact often, but then I have a region here that interacts less often. So somehow you have a walk in 3 space that interacts a lot with itself and then kind of goes away and then interacts again. So it falls back on itself and then goes and then falls back on itself and then keeps going and going. And then you have these different domains, right? So again, trying to find or fit a curve that can reproduce this pattern turns out to be very difficult. All right, so we've been saying that all along that if you have a curve in a confined volume, then the DNA is going to be noted with probability 1. And then this paper came out in Science, people who proposed this method, Hi-C method. And then they said, well, at the megabase scale, chromatin conformation is consistent with a fractal globule. I'll define that in a minute. And it's a knot-free conformation that enables maximal dense packing, right? So I've been telling you that it has to be noted, and here you have Discovery in Science telling you that it's not noted. All right, so let's see what's going on. And this was the cover, which, by the way, is a little bit misleading in the sense that it's a fractal, but it's not this fractal, the one that they study. All right, so what we've been talking about, a random curve in a confined volume, is what they call the equilibrium globule. Is this thing here. And then they proposed the fractal globule, which basically, this is a model that was proposed by Grossberg. And the idea is that somehow the DNA is organized in a way that is self-similar. It's kind of, I mean, a little bit like, it's not quite this, but you can see kind of the patterns that are shown in here. And some of the properties of this fractal globule is that it's non-equilibrium, so it's not something that you withdraw from a Markov chain, right? It's space-filling, it's largely unknotted, self-similar at different resolutions, and consistent with the idea of chromosome territories, as is shown in here. All right, and the reason why they thought this model was better than the model that the equilibrium globule or the random walk model is that when you look at the contact probability of different regions, you see that the random walk will give you this slope, which is about minus 1.5, and the fractal globule gives you about 1, negative 1, right? So here you have, and the observed data is this one, minus 0.9. So here you have your observed data with a given slope, and the fractal that kind of fits very well, and then the equilibrium that deviates quite a lot. So we ask, well, with all the techniques that we have, can we come up with curves that are knotted, hence they are not fractal, that have the same slope? All right, so we would like to say, well, we know this is the random coil, this is the random walk, but can we come up with knotted structures that don't have to be necessarily random that fit this same slope? So this work was done by Rika and Robert, and the idea is, well, this is kind of I was just mentioning. Well, let me go forward. So what we did was to take the simple cubic lattice. This was one of the models that I described the other day, and in which each vertex is located in the, well, has integer coordinates, and points along this walk are at distance 1. And then we use an algorithm called the BFACF algorithm. And the BFACF algorithm, I think people working in our group already know what they are, but it's basically a Markov, generates a Markov chain by these two moves. First, you have this P0 move that it takes one of these corners and will flip it over, as is shown in here. And that it does that with certain probability that we denote P0. And this comes later. Then you have either a P-2 move or a P+2 move. A P+2 move means that if you have a straight segment like this, you are going to replace the middle segment by this little hat here, adding therefore 2 segments. The length increases by 2. If you have a structure like this, you can remove it and come up to a flat place, removing to a flat structure, removing 2 segments. And then this will occur with certain probability P-2. And there is the theory that tells you how to choose P0, P2, P-2, so the algorithm converges and has good properties. All right, so this is what we did. Can we, if we take this, we take any knot, we embed it in the lattice, and then we run BFACF to fill a sphere. In principle, this sphere could be out of equilibrium, could be almost like fractal, but not fractal because, or have similar properties to the fractal, but it's not going to be fractal because we have this type of knot structure in there. All right, so this is how it works. This is a movie made by student Rika. So this is, again, knot plot. As I was telling you, it's good to introduce students to research. So you initially say BFACF, then you allocate the length of your molecule, then you load whatever knot you want to load. These are the probabilities that you are going to accept or reject conformations. You embed this inside a sphere. Now this sphere is going to be filled, so now we empty it, so we just leave the boundary. And now we put these probabilities for the BFACF, and then we run to the length that we want, boom, and then it fills the whole thing, all right? Once we do this, and we generate a conformation. Now we have a conformation in C3, then we need to measure or estimate some type of contact probability. So for every point, what we can do is count 1 for any points that are at distance 1, that is not linearly, that is not consecutive along the genome. And then consider also those points that are diagonally across and just kind of put some weight to those ones, all right? So we tried different methods. The results were all consistent, but this is the one that we reported in the paper. So you can define contact probability by the total, the contacts by the possible number of contacts. Now, this is kind of a technique that you use when you have software that has adjustable parameters. So because now instead of thinking that this is a Markov chain that has 3 parameters, P0, P2, P-2 that are fixed, what we are going to do is take these parameters, -2, 0, the probability of these 3 moves, as adjustable parameters. That means that if I have a data set, then I can try to fit or adjust these parameters so I get the best fit to that data set. In our case, what we want is to fit the slope of that curve that was near -1. So the question is, can I find, for a given node, can I find parameters -2, 0, and +2? So the contact probability fits what I observe in the experimental value. Are there any questions about that? Yes. How do you? Yeah, you are going to do several samples. You take the average of all those. You'll have certain, like a mean and a standard deviation. And then you see this is only the mean. And then you just look for the one that is closer to -1. Any other questions? All right. So what we do at this stage, so for instance, imagine that I have the probability of negative, I take the AND node, the probability of -2 is 0.25, the probability of 0 is 0.25, and the probability of +2 is 0.25. And then I let it run, as I showed you earlier. We do this hundreds of thousands of times or tens of thousands of times. We estimate a contact probability. This average iterations, it tells you kind of how long, how many iterations of how many moves you take to fill the entire capsule, or the entire nucleus of the cell, or the entire sphere. And this is another measure that you take to estimate how compact, how consistent your conformation is with chromosome territories. And this is the number for this particular study, this is the number of conformations we generated. All right, so what we did here, so systematically we start with these values, and then these values will give you this contact probability, and then we start changing P+2. And then see what happens to this contact probability. Then we change P0 and see what happens to this. And then we change P-2 and then see what happens to this. So this is kind of somehow naive way to combine these parameters, but it gives you a good intuition for what might be happening, correct? So what you can expect, for instance, so you understand better what this problem is. So if you put a high P2 and a low P-2, this thing is going to grow really fast. And it's going to fill the sphere very, very fast. If you put a high P-2, but a low P+2, this sphere is going to, or this walk is going to take a long while to increase until it eventually fills the whole thing, all right? So the dynamics can be quite different depending on these parameters. All right, so we found that this combination of parameters, so the probability of accepting the move -2 is 0.25, the same as +2, so they have equal probability, and then 0.40 for just flipping corners gives you this result, and something similar for this other combination of parameters. So now we've done this with the AND node. We fix these values, we fix these parameters, and then we see how are these parameters going to change, or well, how is the slope going to change when I fix the parameters, but I change my node. Okay, so now we said this is the best combination of parameters that we have. Let's see what happens when we change the type of node. So we started with a trefoil, and we have this particular value for the slope, same thing for this. I'll explain you this in a minute. Then we do 5-1, and we get some similar result. 5-2, we get similar result. Now, if you have an entire genome and you have a trefoil or a 5-crossing node, that's not really telling you much, right? So what we want to do is, well, do we have a lot of nodes that are actually affecting, could affect the genome, or at least that it would break this idea of fractal structure. Then we added 20 trefoils, and then we are still within the range of what of this slope. Same thing with 40 trefoils. And then interestingly, the more trefoils we get, the flatter it gets. So it goes away from the random coil. So it becomes more and more organized. Remember that the random coil had a given slope of minus 0.5. This is -1, so this is even less. So this is something we still have to understand why that is happening. So how adding more and more knots kind of gives you a better ordering. And then you ask, what is the percentage of the genome that would be needed? This is a rough estimation, so we can improve these numbers. But what is the percentage of the genome that would be needed to make all these knots? And then you could say that for 20 trefoils, maybe you need about 10% of the genome to be knotted, right? Or to be part of the trajectory of a knot, rather than to be knotted. And the same thing with the 40 trefoils, which you would need about 20%. So you could still have 20% of the genome being involved in these structures, these knotted structures, and having a slope similar to what it was observed in the experiments. All right, so then we went further, and we also noticed that when you look at the fractal globule as published in the science paper, then we noticed that the Hi-C map is actually quite different from what they observe in vivo. All right? All right, so the conclusion from this is that the fractal model, it was a model that was proposed by Grossberg initially, but it was proposed as the best model for the human genome, but this was based only on a single number, which was the slope of this contact probability. Of course, it's consistent with the idea of chromosome territories and so on, but the fractal itself, the fractal model, is the fitting, or is proposed based on this fitting of the slope, correct? The second conclusion that we get is that the fractal model is one of multiple models that fit the data. And it was also another study from a different group kind of proposing that the DNA could be looking like loops. And this is actually the model that people are pursuing now. And some of these models, as we show, can be topologically complex. Okay? So the models that people are pursuing more now is kind of this lariat structure, or the DNA is kind of more organized within these loop structures. Interestingly, people tend to forget, especially in biology, this model was proposed already in the 70s, right? So Lemley extracted DNA from cells. This is kind of a possible scaffold for the cells, and what you get here is this type of loops. So this has been out there for a number of years. But it's now that with these new techniques, we can come back and quantify this and I guess improve what we know, right? But there was this detour through fractal structures. Now the question you might ask is, well, is there any fractal organization of the genome? Well, it might be, but I wouldn't say that this study was conclusive about that, right? So it was the conclusions that they reached was a little bit far. Okay, so then the question that one of the reviewers asked is, well, what do you think? Do you think that we have knots in the genome? Maybe, functionally it could be or could not be. So regions, for instance, that need to be highly condensed, it's possible that they contain knots just by random chance, correct? And regions that are open that need to be transcribed or that are replicated, those regions could be without knots. We don't know that. But it kind of trigger a study done by Koya Shimokawa, Kai Shihara, Rob Sharain, which you've heard of him, but probably you didn't see a picture of him yet. So the knot plot guru, and Maxime, that is somewhere over here. And we look at what is imagine that you have your sphere, and you think, what are the possible realizations of knots within a sphere? Of course, you can think on the standard knot that occupies the whole thing, but you can think that you could take a slab and the knot is actually through that slab. Or you take a tube and the knot is coiled around that tube. So a knot inside a confined volume could have many different conformations. And then we look at, well, out of those many conformations, what's the minimal one? And this has triggered kind of a line of research that basically tries to enumerate the number of knots and the type of knots that you can obtain not the type of knots, but the type of conformations that you can obtain for a given knot and minimal step number. So these studies were already pioneered by Diao in the 90s. He was the first person proving that the trefoil can be made with 24 sticks. Right? Then this followed by Yamaguchi and Ishihara, that they show that the 4-crossing knot has 30 steps and 5-1 has 34 steps. And these proofs are analytical proofs. So you can actually, these are actual theorems. And I don't think we know anything beyond this, analytically speaking. We know through computer simulations, but not analytically. So the way these proofs work is that if you have a knot, then you can project in the plane, and then this gives you a graph, a weighted graph, and these edges tell you the number of pre-images that this edge had. And you know that this G has to fit in a given box in the plane. Then you can generate this this structure. You can try to pull back out of this projection, and then look at the multiple combinations that you can have here. And if you do that search kind of not in an exhaustive way, but kind of in a smart way, knowing how far you can go up and how many edges you can have at each stage, then you can show these theorems. So that's kind of how the proofs go. Again, you take the projection, you get a weighted graph, and then you try to lift up these projections. And this is a paper that we just published a few months ago that tells you what's the minimal step number for knots in a 2x1 tube. So basically you have it's an infinite tube. In this axis, height 1 and width 2. And you can have, this is just one example, but you can have estimates for the torus knots and for the twist knots. And these are, again, analytical results. Now you would like to, this is limited in 2 aspects. You don't get every knot through this process, and you cannot go beyond the 2x1 tube. The reason why you cannot get is because, or the reason why you can do in the 2x1 tube is because you know that it's going to have only height 1, and then you can apply a lot of techniques from 4 plats. So you can think that you are actually doing braids to prove these theorems. All right, so then we, Maxime, together with Rob, work on these simulations. The idea is that we have a knot, and we have the tube, and then what we need to do is feed the knot into the tube and look for the minimal step number of that tube, of that knot. Yes? Yeah.
You consider knot in a, say, in a volume, right?
Mm-hmm.
And then take the trefoil knot, it has this information about the minimum
step number.
Now there is a corresponding information about torsion. Torsion of a curve, you can discretize torsion.
Mm-hmm.
Now, is any result that states the amount of torsion, the maximum amount of total torsion you have for a curve in a confined volume?
We haven't done those calculations, but
you can put bounds on total
yes,
torsion.
yeah,
And because total torsion is influencing energy through torsional energy,
yeah,
you probably have information to control that.
yeah,
Would leave room for bending energy, which is clearly playing a major role there.
yes, yeah, yeah. We could do that. There's studies that have done like this, but I don't think they've been done in confined volumes or for minimal stick numbers. So yeah, we did a little bit. There was one of the reviewers who asked us some more kind of physical questions and along those lines, but we didn't do an exhaustive study.
Because you have torsion discretized on a polygonal
Correct.
lattice,
Yes.
you quickly have a
Yeah.
spectrum.
Yeah, correct. And we have all those conformations in a database, so it would be doable. Yeah, good point. All right, so this is how the algorithm works. We have a tube, we have a knot, and then we want to embed this knot in the tube. So what you do is you first sample knots in the lattice, then you kind of put it in this little box, and now what we are going to see is applying BFACF. BFACF already induces some motion. We're going to see how this knot moves through here. Now, what is going to happen is that the knot, of course, by random motion will move out and in and out, in and out. So what we're going to do is place a wall in the back, so we force the knot to move in one direction. Also, if you notice, this kind of is a kind of a funnel, tends to be wider on this side and thinner, right? And this is the technique we use both for the slabs when we have two planes and for the tubes. Whoops. So let me show you this movie. So we are randomizing. These moves is BFACF. So P plus 2, P minus 2, and P0. So you see that the knot is staying there. This is the length of the curve. And you see how it's being pushed. Once the knot gets here, then it's kind of it's not allowed to go back. This happens with many knots that sometimes get stuck there and there is no way to pull them back or to pull them in, like I showed you there. This can happen for a very long time. There you go, boom, and now you get in there. And now you have to look for the minimal step number of this. So now you let it sample within this tube, right? And as shown in there, and you see this big peak. So somehow you have to get this, when you get stuck, you have to reach out and on the length of the polygon, move out of this region where you get stuck and then get in. And Maxine can give you details on kind of how many iterations this takes and so on and so forth. What is interesting, then we can make these tables where you have the knot type here, then you have the one slab. This was paper published a while back. And then we look at the minimal step number for knots. And some of these, for this column, we can analytically say whether they fit or don't fit. So in this case, we know that this particular knot won't fit on a 2x1 tube. And we also have both the estimates that we get through the simulation and through the analytical results. And in some cases, the analytical results give us better value than the simulation. So our simulations won't always reach this minimum step number that we expect. So there is definitely room for improvement in terms of computational techniques that we can do. And there is also interest in trying to look on how, but we spend time looking at how patterns can increase, correct? So you have the 5.2, for instance, 5.2 has 42, at 5.1, 42 here, 42, and then all of a sudden goes to 62. And different nodes will have different patterns on how they increase the minimal step number as you start confining the node. All right, so another question, and this is the last thing I want to touch on. This is something that we're still in the process of developing. We know that, so we said that, well, you have the genome, and from the genome you can get a heat map, and then people propose models for the heat maps based on the heat maps, on the coiling. But in an ideal situation, imagine you have a node, and then you produce this heat map. The heat map, remember, it tells you the distance in between points. Now you have this, and the question is, well, what can I say? If I look at this, what can I say about the node? Right? And this is a question that comes directly from the biology, and that maybe people have thought about in the past, but we haven't seen any results. So again, this is work with Koya and Kai and Kevin, which is a student of Abby Thompson, who is interested in this problem. All right, so just to give you a flavor, again, another movie, if you have this particular knot in the lattice, you're going to see how you move through this green bar moves, and then how you move through this heat map. And then you try to infer what are the properties of the knot, correct? So you see here, and then you see these two, how one moves with respect to each other. Of course, the diagonal is going to correspond when you are on top of each other, and then the other one goes around. Now you can move to another location and keep going. So then the question is, well, can we, from these patterns that you see here, what can you say about the local or the relative position in between fibers. And there's a lot of open questions in this subject. All right, so let me give you kind of a flavor of what we've done. So you can take a note. This is not in the simple cubic lattice. And if we have some sort of periodicity, at least you can, sometimes you can say an if and only if, but some other times it's just one way. But basically, if you have this kind of wavy patterns that go parallel to this diagonal, then you can claim that you have some sort of periodic behavior. Well, actually, the periodic behavior is a symmetry that tells you, think on the axis that comes out of the plane, and then this knot could be rotated, and the angles at which is rotated corresponds to this pattern over here, right? So it's kind of a fun game, looking for patterns and on knots and the heat maps that they generate and trying to go back and forth. And these are two kind of an example of these theorems. All right, so conclusions from kind of our approach, our studies. We have, I think, new and interesting knot theory questions that naturally arise from the study of DNA packing. In general, polymers. It's also the tube problem also applies to how polymers diffuse through channels, and that has a lot of applications. And random knotting, which is the approach we are taking, provides, or the approach that I'm teaching you, provides an approach for uncovering and suggesting basic rules of chromosome packing. Again, let me emphasize that once we're in the math and we want to go to the biology, we have to be extremely cautious. It's very easy to overestimate the power of the computational tools, all right? So we have to be, and sometimes biology tends to be misleading. It will lead you places that are not necessarily true. All right, and again, just helping the people in the lab. So that's all. Thank you.